Whether we admit this or not, we all like to eat, right? At least I know I do. But with the increase in world population, demand for food crop production is also increasing. Therefore, a farmer's goal is to attain high yield in crop production. Now, because plants need nitrogen for good growth, farmers usually nourish their soils with chemical nitrogen fertilizers. Problem is, these fertilizers are expensive, and when they get into our water sources, they cause water pollution and harm aquatic life. Well, luckily, legumes have a natural alternative of obtaining a natural nitrogen source when they associate with a particular type of soil bacteria called rhizobia. This results in what we call a symbiotic relationship. So what happens is, the rhizobia gets into the roots of the legumes, then specialized root structures called nodules are formed to accommodate these rhizobia. The rhizobia then captures nitrogen from the air and converts or fixes it into a natural fertilizer for the plant. So, what if we could transfer this fixing ability in legumes to non-legume crops such as corn? In order for us to achieve this, we first need to fully understand how the legumes can do this. And this is where my research comes in. So it turns out that the legumes have special molecules called symbiotic genes that give them the ability to recognize rhizobia in the soil, accommodate them in the nodules to fix nitrogen so they can benefit their plant growth. So my research goal is to identify the functions of two symbiotic genes in a legume called alfalfa. To address this, our lab uses a genetics approach. So in the lab, we get seeds in which these symbiotic genes have been knocked out or they've been removed. We germinate the seeds and water their roots with the rhizobia solution to mimic the natural setting. Then after 4 weeks, we check the plants for growth. Results showed that these plants missing the genes did not grow well. They had stunted growth, yellow leaves, and inside the root nodules, rhizobia were dead. So my current question is a dead rhizobia and poor growth caused by the absence of these genes? If yes, my next step is to figure out exactly how these genes are able to achieve this, to keep the rhizobia alive and the plants growing well. Therefore, if we can figure this out, then we can potentially transfer these genes from legumes to non-legume crops such as corn, so that they can obtain this ability and grow well. And because we all need to eat, there'll be plenty of good growing food for the growing population. Thank you.